What are the 4 phases of the cell cycle? Nobody? Nobody knows? There we go. G1 is the first. Yes, Giovanni? Paul, cell growth. So there's 2 phases for cell growth. G1 is 1. What's the other cell growth phase? G2. Good, Tanya. Copies DNA. Good, Danero. Which phase is that? What's the name of that phase? And then M is mitosis. So we got G1, G2, M. Good, Caesar. Synthesis. S is the other phase. So G1, S, G2, M. Those are our 4 phases. Good morning, Fatima. Number 2, what is the relationship between the cell cycle and cancer? What do you guys think? How did we do on the Ed puzzle last week? Did we understand the relationship between the cell cycle and cancer? Can anyone sum it up? You guys can take yourself off mute and speak it too if you want. Or you can type it. I'm going to go over it in a minute, but I want to see if anybody got it first. The cause of cancer depends on the cells. What part of the cells? What did we study last week? One more word, Giovanni. The cause of cancer depends on the cell. Not growth. What were our notes about last week? Or what was the Ed puzzle about? The cell what? What's that other word? What about the division of cells, Giovanni? You got to be more specific. Good morning, Rochelle. Cell cycle. Good, Soraya. So the cause of cancer depends on the cell cycle. All right, I'll take that. Number 3, from your notes last week, what is the difference between mitosis and cytokinesis? Each of them divides something different. It's in that slide about the 4 different phases of the cell cycle. If you guys have your old notes in front of you, it should be there. What do you guys think? Mitosis divides what? And then cytokinesis divides what? Mitosis is the final stage, yes, but what's the difference between these 2 processes, which are both in the M stage? If you guys have a notebook, flip back to your notes from last week. Oops, didn't mean to go forward yet. Mitosis is when the nucleus divides, and cytokinesis is when the cytoplasm divides. Good. So Giovanni, you are correct, but that is the Google answer. I want the answer from your notes last week. Mitosis divides the nucleus, cytokinesis divides the cytoplasm. Good morning, Elizabeth. All right, moving on. Cancer, this is some general information. I read everybody's discussion posts. This is not something you have to copy. This is just something I'm going over because a lot of you are confused about what cancer is. So I wanted to know what you knew. Then you guys watch the video on the cell cycle, which hopefully you learned something about cancer in the cell cycle. A lot of you did not reply to one of your classmates, which lost you points. That is part of the directions. That is part of your grade. So if you got a 6 out of 10, it means you didn't reply to one of your classmates. And you can still go back and do that, and I can regrade it. Just let me know when you do that. So Cancer. Healthy cells have a certain order of growth and division. There's checkpoints throughout the cell cycle to make sure the correct order is followed. So it has to go G1, S, G2, M every single time. The trillions of cells in your body all go through the same exact cycle. If a cell doesn't pass a checkpoint, they're supposed to rest or self-destruct. That self-destruct process is called apoptosis. If these checkpoints are broken, and the cell cycle keeps going, the broken cells keep replicating uncontrollably. If these broken cells stay in one area, it forms a tumor called a benign tumor. If these broken cells get into the bloodstream and travel to other areas of the body, it is now called a malignant tumor, and that is what cancer is. Cancer is the malignant tumors whose cells travel to other sites of the body. So cancer always starts in one place and just forms a tumor. If that tumor breaks off, gets into the bloodstream and travels, that's when it's called cancer. Okay. Another thing people were confused about is the distribution of cancer. Most people thought that cancer is hereditary, and some cancers are hereditary, but maybe only about a quarter of all cancers are hereditary, or strictly hereditary. So 5 to 10% are strictly hereditary. There's a gene mutation that is inherited in the family. Those people have a significantly increased risk of developing that kind of cancer. 10 to 20% is called familial, which it could be in the genes, but there's also environmental factors. So there's an increase of cancer risk, but not as much. But the rest, that yellow portion, which is maybe a little less than 75% of cancers, are called sporadic. And these cancers occur by chance, or they're related to environmental factors like smoking for lung cancer or UV sunlight exposure for skin cancer. So there are more sporadic cancers than there are hereditary cancers. All right. Like I said, if you need to go back and finish your discussion because you only got a 6 out of 10, make sure you do that and let me know so I can regrade it. So essential question, how do cells grow and reproduce? Cells divide during mitosis and cytokinesis. And that is a GIF of the overall process happening under a really, really nice microscope. You can see the cell going through a bunch of different stages and then splitting in the end. We're going to go through each of those stages. A chromosome is one long continuous strand of DNA that is wrapped very tightly around proteins, and the end result looks like an X. So most of you know what DNA looks like. Sometimes DNA just lays around in the nucleus. But when it prepares to divide through mitosis, it has to get thicker and form these structures called chromosomes. The structure of a chromosome. The DNA plus proteins is called chromatin. Half of a chromosome, the left to right halves, is called a chromatid. And when you put them together, they're called sister chromatids. And they are held together at the middle portion called the centromere. Make sure you draw and label the chromosome in your notes. And as we go through the process of mitosis, we're going to be using these terms. We're going to talk about how the sister chromatids pull apart and how this spindle fibers attach at the centromere. So we're going over the structure first, so you know what these words mean as we move along in the process. And as a side note, which I might have mentioned last semester, but I don't remember, the CH doesn't make a CH sound. It always makes a K sound when you're talking about science words. We did mitochondria last semester. There was a CH in there, sounds like a K. Now we have chromosome, chromatin, chromatid. We'll have more as the year goes along. But in science, we don't say the CH sound for some reason. It always sounds like a K. Chromosomes condense or get thicker at the start of mitosis. DNA wraps around proteins called histones, and that is what condenses it and makes it look thicker than it is. So on the left, you can see DNA. That's the picture of DNA that everybody imagines when you hear the word DNA. That picture pops in your head. But when it comes to mitosis, it has to get thicker. So it wraps around the proteins called histones and just keeps wrapping and keeps wrapping until finally it looks like the X structure. And that structure on the right is a chromosome under a really, really nice microscope, one that we don't have here. But labs at research universities have them so they can take really nice pictures. So interphase. The G1, S, and G2 phases of the cell cycle are grouped together and called interphase. This is not part of mitosis. This is everything else but mitosis. Everything else except for the M phase. During this phase, the cell prepares to divide by growing and replicating its DNA. We went over that last week. Gap 1, synthesis, gap 2. And then the picture at the bottom is what the cell would look like in interphase if you actually looked at it under a microscope, under a normal microscope like we have in class. And those pictures are going to be handy when you do your assignment this week and when you do your quiz, because there's going to be microscope-like pictures on both your assignment and your quiz for the week. So you don't have to draw them, but you can always refer back to them. Mitosis and cytokinesis divides the cell's nucleus and cytoplasm in 4 phases. And the end product is genetically identical daughter cells. When a cell goes through mitosis, they are clones. They are the exact same. They're the exact same DNA, the exact same structures, everything. And 99% of your body cells, they perform mitosis. That's why they are the exact same. The first phase in mitosis is called prophase. Chromosomes in the nucleus condense or get thicker. Centrioles and spindle fibers form a kind of net, and the nuclear membrane starts to disappear. So on the left is a nice, pretty GIF animated GIF of how it works. You can see the chromosomes getting thicker and forming the actual X structures. There's the centrioles, and they start to form the spindle fibers. And on the right is a picture that you would find under a microscope if you were in class. It's going to be pictures like that on your assignment and your quiz that you're going to have to pick out. And just this cell here, oops, go back. It's not letting me annotate it. Oh, there we go. Never mind. That's fine, we'll move on. Metaphase. Chromosomes line up individually in the middle of the cell. This is called the metaphase plate. The net, which is the centrioles and the spindle fibers, spreads out and catches the chromosomes at the centromere. Like I said, that's why we went over the vocabulary in one of the earlier slides. That's why I had you draw and label the chromosome, because you should know where the centromere is. It's that middle section of the chromosome. And you can see in the animation on the left, the spindle fibers are pushing all the chromosomes toward the middle to line up. If you look at it under a microscope on the right, it's not that pretty, but you can still kind of tell that the chromosomes are all lined up in the middle. As opposed to the darker ones around it, which are still in interphase. And the cell to the right of it looks like it's in prophase because you can see the chromosomes that are condensed. And the next phase is called anaphase. And the sister chromatids separate to opposite sides of the cell, and they're being pulled by the spindle fibers. So you can see in the animation, they start in the middle, and then they get pulled to the opposite ends of the cell. That is anaphase. And on the right is that picture that you would see under the microscope for anaphase. Telophase is the fourth phase, and it's basically the opposite of prophase. Nuclear membrane reforms, chromosomes begin to uncoil, and the cytoplasm begins to separate. And you can see in the animation, the nucleus is starting to come back, the pink bubbles. The centrioles kind of move away, it doesn't have a job anymore, and the cell is starting to stretch out when it's just going to snap in two. On the right, what you can see under the microscope, you can see it's one cell, but there are two little nuclei forming, and it's starting to stretch out. And it will eventually become two cells. And the last part is cytokinesis. We said last week, cyto means cell, kinesis means split. It can also mean movement, but movement away from each other. So it's the actual splitting of the cell in two. In animal cells, on the left, the membrane just pulls apart and pinches closed. And then plant cells on the right has to form a cell plate because they have cell walls. So that plate eventually becomes a wall.